our next lecture is from Professor Anders Carlsson at WashU. And Professor Carlsson is actually officially retired now, but agreed to come back metaphorically, or at least remotely, and give this lecture to you. I would say he has spent the last couple of years working on a series of lectures, problem sets, and I think a textbook to explain measuring forces and how to be quantitative in your approach to mechanics. He's given a number of these lectures to a CEMB course that we've offered in the fall. And they're just sort of, I just want to say, they're remarkably clear and helpful lectures. So I thought it was very important that you guys got this lecture, particularly at this stage of your career, that you had this lecture, and I'm very pleased that you're going to get it from Professor Carlsson. So Anders, unfortunately, you can't see us, but I can tell you that the students are very good at this point at just yelling out and asking questions. They have had many questions to ask in previous lectures, so I hope they'll do so with this talk. So take the time that you need, but welcome and ask questions.
Okay, thank you, Becky. Yeah, let me second that about the questions. If you let a speaker just go on and on, they will accelerate until you can't possibly understand them. So you need to stop them and get them to kind of recapitulate at some point. And also, any question that you ask, the chances are that other people have the same question. So just go ahead and interrupt. And we're over Zoom, so you might have to be really loud. I hope you've had a good start to the boot camp. I see the list of speakers, and I know these are great speakers, so I'm sure you've been learning a lot of good things already. I'm going to talk, so what would mechanobiology be without force? It's the thing that makes it all work. And I'm going to first talk a bit about general properties of forces in cells, just some things you may not have thought of. And then after that, I'm going to turn to specific kinds of forces and how we measure them. We'll look at molecular motors like myosin and kinesins. We'll look at forces from polymerization. And then we'll look at a little bit larger scale measurements also, like the measurements from whole cells. We'll look at osmotic forces, which are really important, and also at the forces that open channels in the cell membrane. And so, of course, this is a huge topic. I'm going to give you kind of a big picture forest into which you can plant trees. Okay, away we go. So to me, mechanobiology is a feedback loop between force and biochemistry. So let's first think about force. And let me ask you a question. What does a cell need force for? I don't hear anybody yelling yet. Any thoughts about why a cell might need force?
Migration.
Absolutely. Yes. Imagine, for example, that a white blood cell needs to get to a site of infection. It's got to move through a lot of other cells, and those cells are a tissue. So it has to muscle those other cells aside before it can get to where it needs to be. And kind of a smaller scale version of that is cells sticking out pieces of themselves. Some of them are like Philopodia, for example. They stick out pieces of themselves that explore the environment. And cells also need to bring materials into the cell by, for example, endocytosis, and then forces are required to bend the membrane to make that happen. And also, well, part of the job of fungi is to invade, and so fungi need to be able to exert forces to invade other organisms. Um, and something that's been really exciting over the past 10 years is mechanosensing, that cells, by exerting forces on their environment, can sense the mechanical properties of the environment. So this is really happening all the time. And so we have this feedback loop between force and biochemistry. And let me ask you another question. Can you think of an example of how force impacts biochemistry?
Focal adhesion kinases affects the spread of the cell based on the stiffness of the surface on which we are grown.
Okay, so which is the chemical process in that sense that's being affected by the force?
There is activation of the rho GTPases that causes,
Oh,
yeah,
right. Yes, yes, exactly. And so we have the sensing at adhesions, and then something maybe even more remarkable is the fact that downstream, that the force can affect the expression of proteins in the nucleus. And so these are fascinating phenomena. My topic today is more going to be about this leftward arrow at the bottom, about how chemistry gets converted into force. Um, so, and for example, this is we'll be looking at like how polymerization energy is converted to force, or how the energy of solutes in cells is converted into osmotic pressure, those kinds of relationships. Um, so when you think of force, you may think of Newton's second law, F equals ma. And it turns out that that is not really all that relevant in cells, because for the most part, if you look at any element of the cell, the net force on that element vanishes. So we're not thinking so much in terms of F equals ma. If anything, we're thinking that like velocity may be proportional to force rather than acceleration being proportional to force. But it also means that we're often thinking about a stress rather than a net force. So imagine that two of you are holding a rope between you and you're both pulling on it. And that rope isn't going anywhere, but yet there is some force there somehow. And that's the tension in the rope. And any piece of that rope has a rightward and a leftward force being exerted on it, and they're canceling each other. But yet there is a stress in that rope. And that's often how we think about forces in cells in terms of stresses. So here you're seeing on the left is a balance of forces in a circular cell. The internal pressure, the osmotic pressure, is pushing out. But then the membrane is pushing back in. It turns out that internal pressure is tending to stretch the cell, while forces inside the membrane are pulling it back in. So each little piece of that membrane has a balance of forces on it, so it's not actually going anywhere. And if you go now to the middle frame, it looks, this is a little more elaborate. We have a cell sitting on a substrate, and so there are adhesions that are pulling it down. And that adhesion energy would like to expand that bottom area of the cell that's in contact, but that's opposed by the tension in the membrane. So there is a balance of forces. And if you look at either corner of the cell, you'll see that there are these three arrows that are acting on it. Those are forces in different directions, but the vector sum of those forces is zero. And then at the right-hand side, if you have two cells adhering to each other, then it's actually a lot like two cells each stuck to a substrate, but that substrate is replaced by that other cell. So the bottom line is that we're often talking about a stress or a tension rather than a net force acting on a cell. Now, if you're talking about a human being throwing a football, that's a completely different realm. And there, the inertia of your arm, for example, is very important. But at the cellular level, that's not that critical. Okay, and some general facts. Turns out piconewtons are going to be a scale that's important for a lot of the molecular processes. And nanometers are an important length scale. And so it's convenient to have something that relates piconewtons and nanometers. And you can see here on the second line that KT, room temperature times Boltzmann's constant, is about 4 piconewton nanometers. That's something really good to know. So that should be one of your take-homes from this lecture. That's the basic energy scale, the basic force scale, and the basic length scale all combined. When we talk about osmotic pressure, we'll be interested in the scales of pressures, and a megapascal, it turns out, is 1 piconewton per nanometer squared. By way of reference, the pressure in a tire is about 2 atmospheres. And so that's a bit less than a megapascal. So that's the general ballpark of these pressures. This is a unit for both pressure and an elastic modulus, like a Young's modulus. A Young's modulus is really the stress that is needed to deform a material by a lot, say by something like 50%. So here, let's ask ourselves, what is the Young's modulus of Jello? And how would we think about that? Okay, I'm going to go ahead and talk through it, but if anybody is thinking faster than I am, which is very likely, then just go ahead and shout out an answer. But in the meantime, imagine a block of Jello. That's about 10 by 10 by 10 centimeters. So that's a liter of Jello. And imagine how hard you would need to push on it in order to deform it. Well, it's not all that hard. You might need to exert maybe a pound of pressure on it or something like that. Okay, well, if you take that pound of pressure and you convert it to MKS units, and then you divide it by the area, then you find that the Young's modulus of Jello is about 1,000 pascals. It turns out that that's a relevant scale for cells also, that a lot of, especially like the actin cortex, that the elastic modulus tends to be in the range of 1,000 pascals. So essentially, elastically, a cell is a lot like Jello. Now, how are you actually going to measure a force in a cell? You can't bring a scale into the cell and measure the forces. You have to have some relevant length scale or energy scale. And turns out one of those is actually the thermal energy. And we can describe the stiffness of biological filaments in terms and actually measure it using what we know about the thermal energy. So let me show you what I mean. Take a look at different filaments of different stiffnesses. So in this figure, you're seeing kind of a floppy filament on the left and a very rigid filament on the right. And in the middle, it's kind of the Goldilocks filament. And these are all being bent by thermal fluctuations. And the way we quantify these fluctuations is the distance over which the filament changes direction by a lot. So maybe something like 90 degrees. This is kind of a fuzzy definition that I'm giving you, but there is a more precise definition. So there is this persistence length, and you can see that for this middle filament, that persistence length where it's marked, you can see how the direction changes by maybe 60 degrees going along the persistence length. you can see on the left-hand side that the persistence length is a lot less, and on the right-hand side, it's a lot more. And so we can look at a filament that's undulating and describe those undulations via a characteristic length, which is this persistence length. And it turns out that the stiffness of the filament can be quantified by the product of the persistence length and room temperature, which is what you see at the very bottom. So this is a measurement of the stiffness of actin filaments. They're overlapping, and you can see that in that brighter region where they're overlapping, they're actually fluctuating less because they're stiff. And so you can simply watch videos of filaments and from those videos extract their persistence lengths. And for DNA, it's like 75 nanometers. But for actin, it's all of 10 microns. And for microtubule, it's 2 millimeters. So let me ask you another question now. What would the persistence length of a piece of spaghetti be? Say vermicelli, if you happen to be familiar with that. Could anybody guess the persistence length of vermicelli?
Five centimeters.
Okay, I hear a vote for centimeters. Okay, I'll take one more vote just so we have just so we can have a winner. I'll take one more vote. Okay, I guess everybody seems to be on board with the centimeters. Turns out to be 100 light years, okay, which is extremely long. And the reason is that the persistence length scales with a very high power of the radius of the filament, of the thickness of the filament. So it's the fourth power of the radius of the filament. So if you imagine you take the ratio of the thickness of spaghetti and then you compare that to microtubule, you'll get some big number. And when you take that number to the fourth power, you get an enormous number. And that's what gives us the 100 light years. Okay, let's talk about molecular motors. So basically, the molecular motors that we're interested in here walk along filaments. And so what is the basic process needed to do that? What you need is for the motor to stay attached, at least for some time, to the filament, and also it undergoes a cyclic shape change. Okay, so this is actually what this cat is doing. The cat is changing its shape cyclically while it's attached to these stairs. And so that's basically what molecular motors do also. It, of course, looks a lot more complicated once you get the chemistry into the picture. So here is the picture for myosin. The driving the energy comes from hydrolysis of ATP. Okay, well, let's stop there a second. So in hydrolysis of ATP, we go from ADP to ATP to ADP, which means we lose a phosphate. And that releases a lot of energy. But why is that? You know, why can anybody give me a really simple reason why release of phosphate from ATP should release energy?
It's releasing the bond energy that was stored inside the ATP molecule.
Yes, but on the other hand, you know, bond energies tend to be exothermic. Usually it takes energies to break bonds. So what I'm asking you is, why is this now releasing energy? And there's one you know, of course, if you were to actually calculate this number, it would be all kinds of different processes contributing to it. But there's one key thing, which is that phosphates are highly charged groups. Okay? They have charges of like +3, +4, depending upon their environment. And so if you have this phosphate on the end acting with those two other phosphates on the ATP, if that last phosphate can pop off, then it can take advantage of that electrostatic repulsion between itself and those other two phosphates. So it's basically, you know, two positive or two negative charges repelling each other. And in the ATP, it's just in a metastable state that's held together by a chemical bond. Okay, so we start with the ATP. So follow around. It goes 1, 2, 3, 4, 5, 6 clockwise. And in number 1, the ATP attaches. And by the way, this on top there is the myosin. It's a big myosin filament containing many motors, and there's an actin filament below. Then when the ATP attaches, the myosin releases from the actin filaments, and then two things follow: that the myosin changes shape, and the ATP hydrolyzes. You can see there that the phosphate is still in place, then after that, the phosphate is released, and the myosin attaches to the actin filaments and undergoes another shape change, and that moves the myosin along the filament. So I want to emphasize that this particular approach is not universal. Different things can happen during different times in the cycle. Hydrolysis, phosphate release, you know, if you're looking at kinesins, they will be happening at different times in the cycle compared to the myosins. The key thing is that there is this cyclic shape change that's driven by the chemistry. Okay, so we would like to know how much force could be exerted by a myosin motor. Okay, and so we know that the energy available is probably the energy stored in ATP relative to ADP. Turns out if you make an estimate of the theoretical force on that basis, it would be, I think, like 20 to 30 piconewtons. So that's kind of the number we're wondering about. That's the number we're thinking about. Let's see how we can measure this. So this is you notice it's 30 years ago. It's a really classic measurement. It's one of the first papers to show that you could measure these molecular level forces. And it involves a double optical trap setup. So those red cones on the right and left are optical traps, which can hold a bead. The beads are, their energy minimum is in the middle of the trap, and then they can move a little bit to the right and left, and if they're pulled, they'll move a certain distance in one direction. And we know what the spring constant is, so we can, once we observe how much the bead is moved, we can tell what the force is. And the reason we need two beads is just to keep the actin filament stretched. Then below that, we have myosin motors on a bead, and they kind of move their apparatus along until sooner or later the actin filament grabs a myosin motor. And then they can measure the forces as a result of that process. So here we are. Here are the forces. So if you look at the top picture, there's two plots. One is the sideways force, and one is the force along the filament. And you can see that the force along the filament is much larger. And it's intermittent. Every so often, the filament is grabbing on to a myosin, and then it's feeling a force of maybe up to 6 piconewtons, actually, for a few tenths of a second. Okay, so that's our first real number now. We have a few piconewtons for this force scale. And how to think about that? Well, you could compare that to like the maximum force in a chemical bond, pulling a hydrogen molecule apart. If you pulled a hydrogen molecule apart, the force would be much greater than this. So this is small on that scale, but it's roughly on that thermodynamic scale that we would have expected. And of course, there's a distribution of forces. On the bottom, you see that the typical force is like 3-4 piconewtons, and they get out to as much as 6 piconewtons. Okay. So another source of energy to create force in biological systems is the aggregation of different molecules to each other. And so they could, for example, grow filaments. And this is then called a polymerization force. So think of an actin filament or a microtubule. And on the left there, you're seeing a filament and a potential new subunit to add. And then that subunit can add. There is an energy that's released when it does that. That's a very different energy from ATP hydrolysis. This is more like the binding interfaces between the molecules, which might have a lot of hydrogen bonds, for example. And the magnitude of the force is controlled by the free energy of the polymerization as well as the step size. So the step size is the added length of the filament when you add a new subunit. And so for the case of actin, that turns out to be a little less than 3 nanometers. Okay, now there is a really beautiful theory that allows us to extract the maximum force or the stall force from just a very few simple numbers. And those numbers are the step size and also the free energy change upon polymerization. And so if you look at if you look at 1 micromolar actin solution, then it turns out the free energy change is about 2 kT, which is what I have there in the paragraph. And then we simply divide that by the step size to get the stall force, which ends up being about 3 piconewtons. So that's our prediction, that it's actually pretty similar to the myosin, that polymerization of actin should be able to generate like 3 piconewtons of force. So that's the thermodynamics. How does it actually work? And that's probably the mechanism that's occurring is probably something that's called the Brownian ratchet. So if you look down on the left, here's a simulation where the membrane of the cell is moving thermally. It's moving by Brownian motion, hence the Brownian ratchet. And below that, you see a growing actin filament, which is also undergoing Brownian motion, but it's limited by the rigidity of the filament. But the idea is that at some point, the distance between the membrane and the filament gets large enough that a new subunit can sneak in. So here goes. Watch that subunit. Here we go. And the membrane's moving up and down. Whoop, there it went. Okay. And now we're starting over again. So let's run through that one more time. Yep, there it went. Okay. So the growth is actually occurring by a process of fluctuations, and those fluctuations can be either in the membrane or in the filament. And given that, we can actually predict the growth velocity. So on the right, I'm showing a couple of predictions. The solid line is a really classic curve from an analytic theory like 30 years ago. And it showed that there should be an exponential decay of the growth velocity as the opposing force increases. And the bottom points are simulations that I did a little bit later. And you can see that the simulations confirm the exponential decay, but in fact, the whole process is actually a lot slower than one might have expected from the simplest theories. Okay. Now, I'm going to show you our best measurement of the polymerization force of a single actin filament. Notice it's 20 years old. So there hasn't been a lot of progress in that field. There's been a lot of progress on looking at networks of filaments, but we are still pretty limited as for what we know about the polymerization forces of single filaments. And that's because it's very hard to measure. They're just too floppy to get a good hold of them. So this is a really ingenious experiment. It involves a growing filament that is anchored at one end, the red end, by an inactive myosin. And at the green end, it's held by a formin, which is a remarkable protein because it anchors the actin filament to a substrate, but it allows it to grow at the same time. And so what we're going to see is that the filament is going to be able to exert enough force to buckle itself. So here goes. There it went. See, it buckled out. Let's run it one more time. There, it buckles out. So that's what we know. We know the filament can exert enough force to buckle itself. We can calculate that buckling force, and it's about almost a piconewton. So we know that this piconewton could exert almost, or with filament could exert almost a piconewton of force. Microtubules, turns out, are a little bit easier to work with, and because they're more rigid, so we can watch them. Looking at the left here at the top, you see an ingenious arrangement where a microtubule is glued to a substrate and then allowed to grow. Only part of it is glued. The rest of it can bend freely. And if you now look below that at these micrographs, you can see that as you progress from one micrograph to the next, that the filament is growing and bending at the same time. And so we can again use the bending stiffness of the filament to figure out what the force is. And in that way, we can figure out how the velocity of growth depends on the opposing force. So again, we find that the velocity is slowed by the opposing force, and it looks pretty much, it's this exponential decay, just like for actin. But again, the actual exponential decay is a lot more rapid than the theory predicts. In other words, the velocity of growth is actually slower than our simplest theories would have expected. Okay, let's step up a little bit in scale. Let's look at a so-called actin comet tail. On the left, you see, look at the upper left. You're seeing a white bead. That larger white bead has a catalyst for actin polymerization on it that will grow a comet tail of actin. And that tail is being grabbed at the end by a pipette. And on the other side of that white bead, there is a blue bead, which is attached to a cantilever. Okay, so now the force measurement is really going to come from the cantilever, which has a known stiffness. So this experiment works by just letting this actin comet tail grow and then seeing how much this cantilever is deflected by the growth of the comet tail. And in that way, we can measure the force versus velocity again. And now you can see we're talking about forces of nanonewtons. And the reason for that is that we have like thousands of filaments. So then the nanonewton versus a piconewton is a factor of 1,000. So it still means that we're getting like a piconewton per filament. And what's neat here is that we can also measure the velocity for negative forces because the pipette attaches to the comet tail. And so it's found that negative forces will actually speed the velocity of growth. Okay, well, let's get even bigger. Let's look at a whole cell and see how much force the leading edge of a cell can develop. And this is it's an experiment in a similar spirit. Again, there's a cantilever that the cell is pushing on, and the cantilever has a mirror on it, which will change orientation. And so if you shine a beam of light at that mirror, it'll get reflected to a different place when the cantilever is moved. And so that's how the deflection of the cantilever is measured as the cell bumps into it. So this is now a top view of that experiment. Let me go ahead and start this because it'll take a while. So that cell you see on the left is called a keratocyte. And this is one of the real super steady movers. This is the Mack truck of moving cells that people like to look at. It doesn't take a lot of time to decide where it wants to go. It just goes in a certain direction and keeps going. And so it's a good one for this kind of measurement. So you see now it's made initial contact. It's pushing through. It's kind of disappearing just because of the optics of the system. But if you've been watching that green triangle, you can see that maybe it's been moving a little bit as the cell pushes on it. At some point, it makes contact with the nuclear mount, and now it's slipped through. So now it got through to the other side. So from that motion of the cantilever, we can figure out how much force was being exerted by the cell. And what we are interested in here, look on the left first, we're interested in mainly in that little gray area, because that's where the leading edge makes contact with the cantilever. And now on the right, that little gray area is further blown up. And so we're seeing at first as a function of time, there's no deflection until it makes contact. And now it makes contact and the force gets up to like a couple of nanonewtons. Then after that, it drops again. And it turns out that the leading edge figures out that it can sneak around the cantilever. So the cell actually sneaks around the cantilever and then the force drops. But so the total force that the leading edge of the cell can exert is like a couple of nanonewtons. If we look at the whole cell, including the mound around the nucleus and so on, that gets up to about 30 nanonewtons. And the leading edge force probably comes mainly from actin polymerization. When we're looking at the motion of the nuclear mound, then that probably involves both actin polymerization and myosin motors acting to pull up the rear of the cell. Okay, so the take-home message here again is that we get like nanonewton forces from whole cells. Right. Okay. Well, now let's ask a more sophisticated question. Let's not just ask for a total force that is being exerted by the cell. Let's ask how that force is distributed. And actually, let me hide this from you for a second. So let me ask you in general, if you put a cell on a substrate, what's it going to do to it? Is it going to pull in on it, or is it going to expand it? So, well, let's see. I can't see your arms. So if you think it's going to pull in on the substrate, shout, yeah, really loudly.
Yes.
Yes. Yeah.
Okay. Now, if you think it's going to expand the substrate, shout, yes, really loudly.
Yes.
Okay. So it seems to me we had more for contraction than for expansion. And it turns out contraction is the rule. And it's kind of it's a remarkable fact, actually, that in general, it's much easier to generate contraction than it is to generate expansion. And that has to do partly with the mechanical asymmetry of rods like filaments. Because think of an actin filament and think of pulling on it. You can pull on it, and you can pull on it up to like 100 piconewtons, 500 piconewtons, and it won't break. But if you push on it, it'll buckle at very small forces. So it's easier for that actin filament to pull, like it was pulling on those two beads we saw earlier, than it is for it to push on something. And that's reflected in the properties of the actin-myosin network in general, that it tends to pull on things rather than push on them. And so here, then that in general, that gives traction forces where cells are pulling in on the substrate. And so then you see that we have a distribution of forces, and to measure those, one can put fluorescent beads into the substrate, and then follow the motion of those beads. And then if you're really good at elasticity theory and you have a very fast computer, you can convert those motions of the beads into the forces that are being exerted. It's kind of like working backwards. In general, we would have a given force and see what kind of deflection does that imply. So this is doing what we call an inverse problem, which is far from easy, but people know how to do it pretty well by now. Here's a cardiac cell. Upper left, you see it spread out over the substrate, and there's various beads there that are in the substrate that are going to be moving. And moving over to frame B there are arrows that denote, on the one hand, the forces that are being exerted, and on the other hand, the displacements. So what you can see is that the forces are basically radial, and you can't see the direction here from the picture, but it turns out that they are pulling inwards. And one of the really neat things about this is that if you look in frame C, you can add up all those contraction forces. You don't add them as vectors because they would all cancel, but you add their magnitudes, and then this oscillates over time. So this little cell is like a little heart on its own, and the period isn't even that far off from our heart. The other fun thing about this system is that if you increase the stiffness of the substrate, that the cell senses that and then increases the amount of force that it exerts on the substrate. You can also, if you have a good materials facility, you can also put cells on micropillars. And in that case, the experiment is a little more straightforward because the force on each post is just the deflection of that post. And so here is a smooth muscle cell on this array of pillars. And on the right, you can see that for larger spreading for cells that spread more, the forces are larger also. Okay, well, there's a whole nother way of measuring force, which is based on the fact that for certain molecules, we can work out how much force it takes to stretch them. So if you look on the left here, we've got a sensor molecule that can be in two states. It's got a green part and a red part. And if enough force is exerted on it, these red and green parts move apart. And it turns out that changes the fluorescence of the green part, which is pretty natural. Otherwise, if they're close, that red part interferes with the fluorescence from the green part. And this is called a fret sensor. We don't need to get into all the details of why it's called fret. The main thing is that the fluorescence really kicks in when this thing is stretched. So it will fluoresce when force is being exerted on it. And so here this method is being applied to adhesions. The plot you're seeing is red is the force that's being generated, and green is the paxillin concentration in the substrate, in the cell. What this shows pretty clearly is that the paxillin is in the area where the force is being exerted. So this is really saying that this paxillin is an essential part of this force application machinery. And other kinds of sensors based on coiled coils, where these coiled coils can either pull apart or be together and then fluoresce differently, are now being developed. And these are neat because they allow the exploration of a whole different range of forces, and the forces for these sensors can be tuned. And so this was recently applied to endocytosis in yeast. In endocytosis, you need to pull the membrane into the cell. And so this looked at one of the molecules, N4P, which links the actin cytoskeleton to the membrane. And it looked at the forces at various parts of this molecule. And what was a little surprising was that there are different forces in different parts of the molecule. You can see on the left, they're like 8 to 10 piconewtons, and to the right, they're more like 18 to 20 piconewtons. So this means that this molecule is not really just acting in an isolated fashion, just connecting one actin filament to the lipid bilayer. There are other molecules that are interacting with this particular linker molecule in the coat, which is why we get a distribution of different types of pulling forces. Another way of getting a qualitative measure of force, of stress, is just to burn away whatever entity is supporting that stress. So this is a stress fiber on the left. That arrow is where it's going to get zapped. You zap it with a laser, you destroy it, and then on the right, you can see that the ends of the fiber have retracted. And so that means that there was tension in that fiber. Now, if you want to know how much tension, that's a harder question. You'd have to know a lot about the elasticity of the medium. It could be worked out, but the main thing is that this tells us that a tension was present. Okay. There is a huge category of forces that are called entropic forces. And the osmotic pressure, it turns out, is in that category of entropic forces. Now, if you haven't seen entropy or don't remember entropy, don't worry about it too much. The entropy is really the freedom of motion of a molecule in a given space. So if you look on the left, you imagine each of these blue molecules can move in a volume V, which is the volume of the cell. Then the entropy is proportional to the logarithm of that volume. And this is important because in the free energy, which is what determines which way a reaction will go, that entropy appears with a negative sign. So you can see that in the equation for G below here. So that means that there is a lower free energy when the system expands. And you can work out a pressure from that, and it's at the bottom. It's basically the pressure is just kT, our basic unit of thermal energy, times the volume concentration of solutes in the cell. And to remember that, I would just come back to the ideal gas law. The ideal gas law says that PV is equal to NKT, and that's what's on the left there. So this is really just the ideal gas law for the solutes that are in the cell. So how would one measure these? Well, one way that's often used is to start adding solute molecules on the outside, which cancel off the ones on the inside. And so at some point, if you have enough solutes on the if you have as many solutes on the outside as on the inside, then there is no net pressure on the membrane. And so it will start to crumple as we go beyond that point. So basically, the theory is that as we add solutes to the outside, eventually there'll be some qualitative change in the behavior of the membrane at some point. So let's take a look at that. So on the left is what's expected. You first expect a gradual drop in volume, but then there'll be kind of a cusp. And that cusp is occurring at that balancing concentration of solutes on the outside. And if we look at the data on the right-hand side, that's also occurring. This is for budding yeast. And you see these data points, the black circles. You can see that they go down a while, but then there is again a cusp. And that cusp is at the balancing concentration of sodium chloride in the outside medium. And from this, one could infer that the osmotic pressure was almost a megapascal, which is pretty big, 5 times the pressure of a car tire. But there's a big problem with that, which is that the cell is smart. The cell knows what osmotic pressure it needs. And so when you put solutes on the outside, it will generate more solutes on the inside. So that actually confounds the experiment. So this number may actually be too high. And it turns out that there is a faster method which doesn't allow the cell to osmoregulate itself based on nanoindentation. And so it can be shown that if you indent the cell a certain amount, that over a certain region, that the associated spring constant doesn't change. And so you can see that in the right-hand plot. This is now an indenter pushing in on yeast. There's a spring constant, and after about 100 nanometers of indentation, it's a constant. And a physics analysis showed that the resulting that spring constant is related to the osmotic pressure, which ends up only being about 1/5 of a megapascal. So it's only 1 car tire instead of 5 car tires. But that's the general scale of osmotic pressures, that they're in the range of tenths of megapascals, but in other systems, they definitely, like plants especially, they definitely get up to megapascals or several megapascals. Okay, last part I want to talk about is membrane channels and how we might measure their properties. There are mechanosensitive membrane channels, which we think are regulated by tension, although probably it's a lot more complicated than that. But you could think of these channels as being escape valves, that if there's too much tension in the membrane, then they open up and let solutes out, so the osmotic pressure drops. To see how that works, look at the cell here on the right. It's a spherical cell of radius r, and the tension is related to the pressure by the so-called Laplace relation. So anytime there's a pressure inside the cell, there's a corresponding tension in the membrane. And the danger is that that tension gets so large that the membrane tears. And that might be what these channels are there to prevent. So we'd like to know, how large a tension do you need to open those channels? And what you do is simply to apply a pressure to the cell via pipette, as you can see on the left. And then if you look at the right now, there's a very fine line in the middle, kind of hard to make out, but it's there. You can see that as you go from left to right, that line goes from straight to being curved. And that means that there's a pressure that's been applied, and the tension can be calculated from that pressure using the Laplace relation. So we're able to know the tension as we apply this pressure. And in that way, we can get the opening probability of a given channel as a function of tension. So here, it's a particular channel called MSCS. You can see that it opens up pretty suddenly at around 5 or 6 dynes per centimeter. And for other channels, it might be in the range of 10. But so that's how we measure these, and that's the general ballpark. I want to mention this is something exciting I just came across yesterday, actually, that a recent measurement, a way of measuring absolute osmotic pressures based on double emulsion droplet sensors. And these are droplets which they consist of a relatively thick layer of lipids. And inside they have a well-controlled medium, which is basically water, and an osmolyte, which is, I think, polyethylene glycol in this case. So you have these well-characterized droplets. And in the lower right, you can see how they're calibrated. You can see that their pressure, that their volume drops under external pressure. And then you can pop these into cells and actually measure the absolute osmotic pressure in the cell. So this is pretty remarkable. I would say the main trouble now is that they're kind of on the large size. So they only work for really large cells, but still this is a really promising direction. So I'll call it quits there. We are kind of, this is still a field that's in progress, measuring forces in cells. It's kind of that we every so often we find a really ingenious method and we're able to measure a new kind of force, but it's far complete at this point. But new technologies are always coming about, and we are now beginning to be able to measure the stresses inside cells. So I'll call it quits there, and I'll be happy to take any questions.
I had a question regarding the ablation method you mentioned.
Yes.
When you use ablation, do you ever run the risk of maybe destroying other parts of the cell that you don't want to destroy, and does that affect your experiment?
It's not that, I don't think that's that big an effect, and the reason is that it's a very localized kind of thing, that you can actually create a very fine laser spot, and you can even make it so that it only focuses at one point in 3 dimensions. So you might worry that there would be damage above. On the whole, that is not that big a concern. I've seen this used for measuring a lot of things, evaluating stresses during a lot of different kinds of processes, and it's only being done for a very short time. Of course, you have to calibrate it. You're going to want to use the lowest possible dose that you can have that breaks the filament of interest. But my understanding on the whole is that is not a big problem. Thank you.
Thank you.
Yes. There was this slide you have about measuring the force due to actin, and you have a pipette and a cantilever. So the question that came to my head is, the cantilever is touching the actin at one end and the pipette on the other end. How is the actin polymerization going on if there are 2 contacts?
Ah, right. Okay, let me go back to that picture. Okay, yeah. So I think you're wondering how the polymerization can proceed with that pipette attached. And it turns out that in this case, the polymerization is actually occurring at the white bead. So the white bead is coated with an agent that nucleates actin filaments. They go by various names like WASP or ACT-A. And these are agents that activate the ARP2/3 complex to form new filaments growing on the sides of existing filaments. And remarkably, they are able to grow right there at that interface. So that's actually a characteristic of actin polymerization in cells in general, that when you look at these actin networks in cells, they tend to grow from the membrane. So you can actually think of the membrane as a filament factory. So it's completely possible for these new filaments to develop while the whole gel is still attached to the bead. So is that answering your question?
Yes, please. Thank
Yep.
you.
Okay. Mhm.